This is part 2 of the lecture on the lymphatic system and immunity. At this point, we will talk about body defenses. Body defenses basically is the way in which your body identifies and protects itself against pathogens. What could pathogens be? Well, they could be anything that can cause a disease, as listed here: bacteria, viruses, other complex organisms, some protozoans, some spores, some parasites, some fungi. In addition, body defenses protect people against foreign toxins. There are many pathogens that make toxins. Also, toxins can be accidentally or intentionally ingested, and your body has defense mechanisms against those. Your body defenses are divided into two groups. It's divided into your innate defenses or non-specific defenses. These are generalized defenses. They protect against many different things. We will talk about these first. The second type of defense mechanism are called adaptive defenses. Adaptive defenses basically mean immunity or protection against a specific foreign substance that you've been exposed to. These are carried out by lymphocytes and helped by monocytes or macrophages. These are the different types of non-specific defenses we're going to talk about. Species resistance, mechanical barriers, chemical barriers, natural killer cells, inflammation, the inflammatory response, phagocytosis, and fever. They're all interrelated, but we'll talk about them individually. I mentioned mechanical barriers first because the skin and the body's mucous membranes, the oral mucosa, the lining of the bladder, those are all mechanical barriers against foreign invaders. Mechanical barriers such as the skin are considered the body's first line of defense against foreign invasion, which is why I mentioned them first here. Mechanical barriers can include such substances as tears and saliva that basically wash away as the tears flush across your cornea of potential foreign material. Saliva obviously attaches to foreign material and helps press it make its way into the stomach, where it can be destroyed by gastric acid. Urine is also a way of washing away potential pathogens that line the mucous membranes of the bladder. So anything that helps to protect the body to avoid getting through the skin is considered a mechanical barrier, the skin being the most important mechanical barrier we have. Species resistance. Species resistance basically means that certain types of species are susceptible to certain types of foreign invaders, and other species are not susceptible to that foreign invader. So each species has unique receptors that foreign invaders have devised to allow them to infect or to become attached to or to grow in or on certain cells, and different species have different receptors on their surface. So for example, you can get strep throat, but your dog can't get strep throat. Your dog can get distemper, but you cannot get distemper because you are a different species from the dog. You have species resistance. HIV or AIDS alters species resistance, altering your body's immune system and allowing the body to be infected by normally resistant organisms, normally organisms that your body would not be susceptible to due to species resistance. The next non-specific defense mechanisms are chemical barriers. Chemical barriers are substances in body fluids that provide a chemical barrier to foreign material. And they include gastric juice with the low pH, the high hydrogen ion concentration, an enzyme called lysozyme in tears, which has an antibacterial function. Salt that's present, sodium that's present in sweat. Those are all chemical barriers that are produced by body cells to fight directly bacteria. Also, substances like interferons. Interferons are small peptide molecules that are secreted by, in many cases, infected cells to protect the cells around it, especially cells that have been infected by viruses. They also stimulate phagocytosis in cells that are infected. Defensins. Defensins are also small peptides. These are produced by neutrophils primarily, and they are basically substances that are produced by neutrophils that perforate or break down bacterial cell walls. Remember that bacteria have not only a cell membrane, but outside of that they have a cell wall that our cells do not have. And perforation of that bacterial cell wall will kill that bacteria. They don't affect our cells because our cells do not have cell walls. Penicillin is another antibiotic that basically affects and destroys bacterial cell walls. That's how it works to kill bacteria. Collectins are also small proteins that are effective both against bacteria, yeast, and even some viruses. They are substances that are secreted by cells of the immune system, but in a non-specific way, and they bind to carbohydrate molecules that show up on the surface of infected cells, and when they bind to these, they clump the cells together so they're more easily attacked by the body's immune system. And the complement system is the final chemical barrier that we're going to talk about. Complement is a group of proteins that basically float around in inactive form in your plasma until they are activated. And when complement is activated, it's very similar to the activation of the clotting cascade. Basically, one complement protein activates the next, and ultimately, the activated complement proteins do several things. One of the things they do is they enhance the phagocytic ability of certain white blood cells like neutrophils and monocytes. They also increase your body's inflammatory response, which is going to call more cells to the site of the infection, and they call those phagocytes to those areas. This is a diagram showing the complement system, and again, it's a series of proteins. You don't need to know the specifics. What I'd like you to know about this is that when the complement system is activated, what happens is it basically recruits or calls white blood cells to the site of that inflammation. So it calls them to the, it does directly kill them by clumping them together, and it clumps them together so that phagocytosis can be more effective. Natural killer cells. Natural killer cells are a small population of lymphocytes in your circulatory system that basically defend against viruses and sometimes some cancer cells. And what they do is they secrete substances called perforins. Perforins are substances that perforate. They basically destroy the affected cell's wall and therefore kill the cell. Perforins are produced by these natural killer cells, but not to a specific foreign substance, but to many different foreign substances, which is why it's considered a non-specific form of the body defenses. It represents about 7% of your circulating lymphocytes. This diagram shows this is a cancer cell here, which has special markers on their surface, and when one of these natural killer cells, which is floating around, identifies that natural killer cell, identifies that cancer cell, having that specialized marker on its surface that normally aren't there on cells, it secretes the perforins, which then kills the cell. Many of the studies involving cancer treatment at this point involve trying to increase the number or the activity of natural killer cells. Because they would treat by just attacking the cancer cell and not attacking your normal cells that don't have that cancer antigen or foreign substance on the surface. Inflammation. Inflammation is another very, very common non-specific body defense mechanism to infection. Whenever you are asked to think about inflammation, think about what happens when you're stung by a bee. When you're stung by a bee, bee venom is inserted under the skin, and the first thing that happens is you get swelling, redness, warmth, and pain. Swelling, redness, warmth, and pain are the characteristic signs of inflammation. The swelling is because the capillaries become leaky. The capillary permeability increases, so increased fluid leaks out of the capillaries into the surrounding tissue. The blood vessels dilate, increasing blood flow to that area. That's what causes the redness. The capillary permeability increase causes the swelling. And as that fluid leaks into the tissue spaces, it pushes on the cutaneous nerves under the skin, which causes the pain. And obviously the increased blood flow causes the redness. So those are the classic signs of inflammation. It's a non-specific response to any essential foreign substance. Could be infection, could be a foreign body like a splinter. In addition to that, other parts of the inflammatory response include invasion of white blood cells if there's infection. If it's due to an infection, white blood cells will be called to that area, and then the tissue fluids with clotting factors will seep into there. Fibroblasts will arrive. Those fibroblasts will start creating a connective tissue capsule around that area of inflammation to basically wall it off. Your phagocytes, your neutrophils, your monocytes and macrophages will be active in increasing destruction of and removal of foreign material or bacteria, and then ultimately healing will occur, the normal cells will divide. So those are the actual actions involved in the inflammatory response and what you see. Phagocytosis is part of that. Phagocytosis is that process of removal of foreign material, they could be living or non-living, from the lymph, from the blood, from the tissues. Phagocytes are present not only in the blood vessels and in the lymphatics, but also in the lymphatic organs such as the spleen, the liver. They are present in the lymph nodes. Some macrophages basically set up shop in specific organs and sit there waiting for foreign material to occur. This makes up what's called the reticuloendothelial system or the mononuclear phagocytic system. And various organs of the body have their specific cells that live in them. For example, when we talked about the neuroglia of the brain, we talked about the microglia being the scavenger cells of the brain tissue. We have fixed macrophages within various organs, including the liver. There's fixed macrophages in the spleen and other organs of the body, just waiting for foreign materials so that they can ingest it and destroy it. Fever. Fever is also a non-specific reaction to some type of infection or inflammatory reaction. It occurs usually when a viral or bacterial infection occurs, stimulating T lymphocytes, which are in part of the adaptive or immune response, to produce a series of chemicals and secretions. These secretions are collectively called interleukins. One of those, interleukin 1, which is also used to be called endogenous pyrogen, is basically a substance released by the T lymphocytes that then binds to receptors in the thermoregulatory center, an area of the hypothalamus that's your brain's thermostat. And what it does is it sets your brain's thermostat up higher and you develop fever. And the question is, why does your body do that? Well, your body does that because as your temperature goes up, a couple of things happen that help to fight off the foreign material. One of which is that phagocytosis is increased at a higher temperature. The second thing is that when your temperature goes up, the liver and the spleen to some extent, but mostly the liver, increases its sequestering of the element iron, which is found generally floating throughout the bloodstream, and your iron levels in your blood go down. When the iron levels go down, bacteria and fungi that need iron to grow don't get as much of it, so they don't grow as much. So the next time you get a fever from whatever infection you may have, remember that your fever is actually being produced by your body for reasons listed here to help fight the foreign material or foreign infection that caused the fever. So that's an overview of the non-specific, or what's called the innate defenses, and what they do. The rest of today we will talk about the adaptive or specific defenses, which is also called the immune system. The immune system means the development of foreign attack to specific foreign substances that you're exposed to. To do that, you have to know what's you and what isn't. It's based on the idea that you have to be able to identify what is yourself from something that isn't yourself. Anything that isn't yourself or is non-self can be considered an antigen. In other words, it is recognized as not of your normal self, and therefore can be identified and attacked by your body's immune system. What is an antigen? Well, antigens can be many things. They can be proteins. They can be polysaccharides. They can be glycoproteins. That's proteins with carbohydrate groups on them, glycolipids. Most effectively, an antigen has to be large so that your body's immune system can see it. Some antigens are too small for your body's immune system to identify. Those are called haptens. Haptens are molecules that are not antigenic by themselves, but when they combine with a larger molecule, they can stimulate an immune response. For example, penicillin in and of itself is too small a molecule for your body to identify, but when it binds to a substance in the body that makes it more identifiable, then your body can react to it, as people with penicillin allergies very well know. So a hapten is a small molecule, too small for your body's immune system to find as antigenic, and there's no immune response. But when haptens enter the body, they can bind to carrier molecules, and when they do that, all of a sudden then they're clustered together and they make up a complex that's big enough that can cause an immune response and cause your body to identify those and then mount an immune reaction to them. To understand the process of developing an immune reaction, we have to understand the basic cells associated with it. Remember that all of your blood cells, your red blood cells, your white blood cells, and your platelets, all in the adult stage develop in the bone marrow. The bone marrow produces the monocytes that will then give rise to the macrophages, releasing them into the bloodstream. The bone marrow will also develop the two forms of lymphocytes, the B lymphocytes and the T lymphocytes. The B lymphocytes, when they mature, which will happen in the bone marrow, are then released into the bloodstream, where some of them float around in the bloodstream, others seed themselves into those germinal centers of the lymph nodes and sit there waiting for foreign material to come by. The T lymphocytes are also produced in the bone marrow, but when they're released, they're not mature yet. They then have to be taken by the blood to the thymus gland. That's that gland we talked about in the mediastinum, where they mature. The thymus gland is the site of maturation of the T lymphocytes. The bone marrow is the site of the production of the T lymphocytes. Once the T lymphocytes are matured, they are then released from the thymus, where they also float through the bloodstream, and they also seed themselves inside lymphatic organs like lymph nodes, like the spleen, and so forth. The immune response is broken down into two wings, the cellular immune response and the humoral immune response. The cellular immune response involves only T cells or T lymphocytes. And for the cellular immune response to occur, the T lymphocytes, which are made each for activation to a specific foreign substance, have to be presented that foreign substance. In other words, a T lymphocyte that is specific for a specific antigen, if it comes across that antigen, it will not activate itself. But it needs to be presented, that antigen, by what's called an antigen presenting cell. Antigen presenting cells are largely macrophages, although some B lymphocytes can do it. So a T lymphocyte will float around until it is presented an antigen from a macrophage. And when it's presented with that specific antigen that's specific for that T lymphocyte, then that T lymphocyte will be activated, and we'll talk about what happens there. How does that antigen presentation occur? Well, it's based on the fact that all of the cells of our body have a specific group of antigens on their surface that identifies them as us. Those are called the histocompatibility complex or the human leukocyte antigens, HLA or MHC markers. These are markers, proteins on the surface of our cells that identify these cells as ours. T cells, when they're activated, produce secretory substances called cytokines. We've talked about one cytokine when we talked about interleukin-1 being produced to cause fever. Once a T lymphocyte is activated by presentation from a macrophage of an antigen, it then starts to divide, and it forms many, many clones of itself. And then as those clones form, some of those T cells become helper T cells, some become cytotoxic T cells, and some become memory T cells. For this cheat chart, I just want you to look at the top two. The bottom two are beyond what we're going to do here. Basically, there's MHC proteins on the surfaces of the cells. There's two classes of those. There's class 1, which are found on all nucleated body cells. Every cell in your body that has a nucleus, which would obviously abstain red blood cells, which do not have nuclei. But all the other cells in your body have these class 1 markers on their surface. So when they talk about finding a tissue match for a transplanted organ, they're going to look for as close a class 1 match on the surface of those cells. Class 2 markers are only found in certain cells, generally only antigen-presenting cells. What antigen-presenting cells are those? Certain B cells, but primarily macrophages. These are the cells that are going to be identifying the foreign material and then presenting it to the T cells so that they can form a T cell response by activating that T cell. So once a T cell is activated, it can become several things. One of the things it can become is a helper T cell. Helper T cells are some of the clones of the T cells that are activated. They're also called CD4 cells. And we'll talk about how they work to stimulate the production of cytokines that activates B cells and to make antibodies. The other type of T cell clone would be a cytotoxic T cell, which are called CD8 cells. These cells basically cause direct death of the infected cell by the production of that perforin protein that destroys the infected cells. That's why they're called cytotoxic or cell-killing T cells. And then the third population of T cells, when a T cell is activated and starts to divide, are memory cells. They don't do anything to the initial exposure, but if that antigen represents itself at a later time, they will differentiate very quickly into cytotoxic and some helper T cells. HIV or AIDS is a disease of helper T cells where they are not functional and causes the condition associated with that. So this is a diagram that shows the immune system, and what we're going to be looking at in this part is just this half of it. We're not going to be looking over here yet. This is the B cell related, or the humoral part. This is the cell mediator, and it begins with an antigen presenting cell. This is a macrophage right here, and this macrophage is ingesting some foreign material, maybe a bacteria, and when it does that, you have these antigen, these MHC markers on the surface like we saw on the last slide. These class 2 markers are sitting on the surface of that cell. And what happens is that this macrophage will ingest that foreign material, digest it by fusing it with lysosomes, and then take antigens from that and present it along with those MHC2 markers on the surface. And when they do that, the T cells, which are floating around and can't do anything until they're presented the antigen, then can recognize that antigen on the surface of the antigen presenting cell and become activated. So activation of the T lymphocyte occurs on presentation of the antigen by the antigen presenting cell, which we will say is almost universally a macrophage. When a T cell is activated by this presentation, the first thing it does is it starts to divide by mitosis. It forms 2, then forms 4, then forms 8, then forms 16. And some of those clones of that activated T cell are going to become T helper cells, and they're going to secrete cytokines, which are going to activate B cells that we'll talk about in a little bit. Some of those T cell clones are going to become memory cells, and they're going to basically wait for the next time this shows up. And some of those activated T cells are going to become cytotoxic T cells. Cytotoxic T cells will directly attack other foreign invaders like this and destroy it by producing that perforin substance we talked about. So an activated T cell, when presented with an antigen by an antigen presenting cell, starts dividing and forms lots of clones of itself. Some of those are going to become T helper cells, secreting cytokines, including interleukin-1, which gives you a fever, non-specifically, but also cytokines which are going to activate B cells to form antibodies that we'll talk about in a few minutes. And other clones are going to become cytotoxic T cells to directly attack more of this foreign material. And some of them are going to become T memory cells that are going to sit around and sit this one out, but wait for the next time infection by this specific organism occurs, and then they can react very quickly, causing what's called the secondary immune response. Those cytokines that are produced by the T helper cells come in several different types. The so-called colony stimulating factors. If you think about that, colony stimulating, that means making more cells. Basically, this stimulates the bone marrow to make more lymphocytes. So if your body is fighting an infection, one of the things you're going to want to do is you're not only just going to want to fight with what you got, you're going to want to call in more troops. So the colony stimulating factors are going to basically call in more troops, pull in more lymphocytes, tell the bone marrow, let's make more lymphocytes. Other cytokines are what are called interferons. Interferons are a group of substances that block the replication of viruses. Viruses are substances, non-living substances, that alter an infected cell's DNA to make more viruses, much like what we're going through here now with the COVID-19 or coronavirus. Interferons are one of the classes of drugs that they are studying to try and see if they can treat this infection. Interferons also stimulate macrophages to increase phagocytosis of those viruses, and they stimulate B cells to produce antibodies, and they directly can attack cancer cells. So interferons is another class of cytokines. Interleukins is a third type. We've already mentioned one type of interleukin, interleukin 1, or endogenous pyrogen, that causes fever. Other interleukins cause the differentiation of lymphocytes into different types and the growth of the colony of lymphocytes to help increase lymphocyte activity. And finally, tumor necrosis factor. Tumor necrosis factor is a substance, a cytokine that's released by activated T lymphocytes that works to try and stop the growth of tumors. It also releases other growth factors. It can cause fever. It also causes an increase in your body's inflammatory response. And it stimulates lymphocyte differentiation into more aggressive types to fight infections. Why are these cytokines important? Well, they're important because there's a lot of medications out there now that affect these. For example, colony stimulating factors stimulate more lymphocyte production. They make more white blood cells in the blood. And sometimes you want to do that. For example, people with certain cancers that have chemotherapy, that have low white blood cell counts, you can give them synthetic colony stimulating factors. For example, you can see on TV, sometimes they talk about commercials for Neulasta. Neulasta is a man-made form of a colony stimulating factor. And what does it do? Well, it basically increases your number of white blood cells. This is used for people who are taking especially certain chemotherapeutic regimens where their white blood cell count's very low, or people that for other reasons have very low white blood cell counts. This is a drug very commonly used to increase your white blood cell counts. Tumor necrosis factor inhibitors. Well, we said that tumor necrosis factor stops tumor growth, releases growth factors, and it causes fever, and, which isn't mentioned on here, it causes inflammatory response and increased cell division. So why would you want to block that? Well, you want to block that in cases in which you have conditions where inflammation and abnormally heavy cell division is occurring. But you've got to use that in somebody who doesn't have any tumors, because if you block it in somebody that has tumors, you could make them worse. Tumor necrosis factor blockers help stop that inflammation that is caused by the tumor necrosis factor. This inhibits that. They're used to treat rheumatoid arthritis, which is an autoimmune disease that causes inflammation, juvenile rheumatoid arthritis, psoriatic and plaque-like psoriasis and arthritis, other diseases like ankylosing spondylitis, ulcerative colitis, and Crohn's disease. Basically, they work by blocking tumor necrosis factor. So they work to do what they do to make the body heal, but they also raise your risk of infection. A number of these are on the market. You don't need to memorize these, but I bring this up because you can't look on TV without seeing commercials for Humira, for plaque psoriasis, Enbrel. You'll see Remicade. A number of these drugs are very, very commonly used for these conditions, very oftentimes advertised for on TV, and this is what they do. They block the tumor necrosis factor, blocking the inflammatory response, and blocking the abnormally high cell division that causes plaque-like psoriasis, and make those symptoms get better, but with the risk being you lose the inhibition of tumor growth. So obviously, if you have any type of a tumor, you will not want to be on this class of drugs. The last aspect of the immune response is called the humoral immune response, or the B cell response. We haven't talked about the B cells at all. We said before, B lymphocytes are produced in the bone marrow, released into the bloodstream. Some of them float through the bloodstream. Some of them adhere to those germinal centers in the lymph nodes. They also adhere to the white pulp of the spleen. And they have on their surface antibodies, each one to a specific foreign substance. And they are activated by the presence of that foreign substance and the cytokines released by the T cells when they're activated. So a B cell just coming across an antigen that it is responsive to is not going to be activated unless it receives the cytokines from the T cells that then activate it. Once a B cell is activated, just like a T cell, the first thing they do is they start to divide. That cell becomes 2, which becomes 4, which becomes 8, which becomes 16, and ultimately those huge number of cells that are going to be cell clones that are going to be produced by that one cell are going to divide themselves into 2 groups. One group are going to be plasma cells, which are going to produce and release antibodies into the circulation that are against that foreign substance. And the others are going to be memory cells, much like the T memory cells, which are going to sit this one out, but they're going to wait for the next time that foreign body occurs, and they can give a more aggressive response to it. Antibodies are also called immunoglobulins. These are those gamma globulins we talked about when we talked about the substances found in the plasma. So at this point, we are going to now look to this side of the chart. This is a B cell, and it has these Y-shaped structures, which are antibodies on its surface. And these antibodies are to one specific antigen. So each B cell is only responsive to one specific foreign substance. And if that foreign substance is around, that antigen can bind to that antibody. But even then, that B cell is not activated unless the antigen binds and it receives cytokines from an activated T cell to that antigen. And once those two things occur, then that B cell becomes activated and it starts to divide to form B cell clones. This is another diagram showing different types of B cells with different specificities on their surface. For example, this B cell has antibodies on its surface for an antigen that looks like this one for an antigen that looks like this one for an antigen that looks like this, and this one for an antigen that looks like that. So if an antigen looking like this comes along, this bacteria with this marker on its surface, this one will not activate because it doesn't bind to it. This one, it will not bind to this one either. This one, it will. And when this antigen binds to this antibody on the surface of this B cell, in the presence of cytokines from an activated T cell, this activated B cell now produces B cell clones. 2, then 4, then 8, then 16, then 32, then 64. So to activate a B cell, it has to have the right antigen receptor antibody on its surface for that antigen, and it has to receive the cytokines from an activated T cell. And once those two things occur, then that B cell is activated and it starts to form those B cell clones. What do those B cell clones do? Well, those B cell clones are going to divide into 2, 4, 8. In this case here, this one's dividing into 2 and then dividing into 4. Of those 4, 2 of them are going to become memory cells, and they're not going to do anything. They're going to sit, but then the next time this antigen comes around, there's going to be a more aggressive response because you'll have all of these already built up. The other 2 are going to enlarge. Their rough endoplasmic reticulum is going to become very dense, and they are going to produce antibodies as plasma cells. They're going to produce substances like this, but they're going to release them out into the circulation where they are going to bind to foreign antigens that's around. So these are released antibodies that are released from these plasma cells, from the activated B cells, and they're released with antibodies that are specific for that specific antigen. Antibodies basically are complex protein molecules, consists of two what are called heavy chains and two what are called light chains of proteins, and they form a Y-shaped structure where on the ends of these two ends have what's called the variable region. These are the regions that are responsive to a specific foreign antigen. In this case, it would be a round one. In the last slide, here it was a pointed one. In other words, a specific antigen is made on the surface of the antibody. So these are the antigen binding sites. And then this is what's called the fixed end here. So how do antibodies work? Well, antibodies basically don't really kill antigens. What they do is clump them together. So here you have a whole bunch of antigens floating around, and you have a bunch of antibodies specific for those antigens. Because those antibodies can bind to two antigens, what happens is you end up with a clumping of those antigens and the antibodies, forming what's called an antigen-antibody complex. And once you've clumped this together into this complex, it's easier for your body's immune system to destroy it. The phagocytes can chew it up a lot faster because everything is clumped together. Here's another example. This is an antigen here, and these are antibodies that have clumped this big green antigen together into this mass so that it's clumped them together so that they're easier to destroy by your body's natural ways. What kinds of antibodies are there? Well, there's 5 different classes of antibodies. IgGs, IgEs, IgDs, which look like what we've talked about, IgMs, which look like 5 of these hooked together in a circle, and IgAs, which look like 2 of these hooked together end to end. But they all have the same thing. They all have this variable region here, which has a specificity for one specific foreign substance. This one has all these that are made for the exact same foreign substance. This one has 2 for foreign substance, and 2 and 2. Where are they found? Well, the IgGs and the IgMs, basically float through the bloodstream. These are the antibodies that float through your bloodstream looking for antigens to bind to and clump together. So if we go back to here, the IgGs and the IgMs, these are the ones found floating in the bloodstream. IgAs are found in exocrine gland secretions. What does that mean? That means they are found in secretions of the intestines, secretions of the GI tract. They're found in tears. They're found a little bit in saliva. These are antibodies that are found in secretory regions of the body. IgDs are those antibodies found on the surfaces of the B lymphocytes. Remember, if we go back and look at the B lymphocytes again, we said that the B lymphocytes have these antibodies on the surface with the antigen. These are IgDs. So the IgDs are the antibodies that are stuck in the membrane of the B lymphocytes. And then IgEs are the antibodies that are found on the surfaces of basophils and mast cells that are involved with allergic reactions and inflammatory response. How do antibodies destroy foreign material or cause foreign materials destruction? Well, we said the first thing they do and the most important thing they do is agglutinate them. In other words, they take the antigens and they clump them together. When they clump them together, they're more easily attacked. What else do antibodies do? They precipitate. When they clump things together, some types of antigens are actually dissolvable in tissue fluid. They come out of solution, become solid substances, which are, again, easier for your body to phagocytize and destroy. The other thing they can do is neutralize foreign toxins. There are sometimes foreign invaders make substances that are toxic to the body, and those toxins can have antibodies made to them, and when the antibodies bind to the toxins, they lose their toxic properties. The example for that is there's a organism called diphtheria. It's a bacteria that we don't see anymore because everyone gets DPT vaccines when they're kids. But diphtheria, if you got it, would the infection, the diphtheria organism would cause the development of a toxin, which could be very dangerous to the body. But your body's immune system can make antibodies to that toxin to make it not life-threatening. So the direct attack this is the way in which antibodies work. Antibodies also activate that complement system that we talked about. That complement system that basically causes, again, the collection together of foreign material, much like agglutination. They also cause the chemotaxis, the pulling of macrophages and neutrophils to the region. It causes the direct lysis of some bacterial cells, neutralization of foreign material, and activation of your body's immune system. Antibodies also can have localized changes by causing the inflammatory response to occur, which is why some people, when they get vaccines, will get an allergic or an inflammatory response to them. This is a slide just kind of basically going over the steps in antibody production. Obviously, the T cell is what it starts with, and it basically has to be activated, releasing the cytokines, and then when those cytokines are released, the B cell, which has encountered the antigen that it's specific for, meets up with those cytokines, and then it's activated to divide and produce many, many clones of that B cell, some of which produce antibodies as plasma cells, others become memory cells. So if you think about the last time you had strep throat and you had swollen glands, why were those glands swollen? Well, those glands were swollen because the antigen from the strep had traveled through the lymphatic vessels to those lymph nodes. And when it reached those lymph nodes, it activated both T and B lymphocytes in those lymph nodes that were specific for that antigen. They formed huge numbers of clones of themselves, which caused the lymph node to swell, which is why your lymph nodes swell when you have your body fighting an infection. Immune response. Immune response can be of two types. A primary response means your body has never been exposed to that antigen before. And when that happens, you have no memory cells. So if you are exposed to a foreign material and you have to develop an antibody response, it's going to take about 3 weeks to get a maximum antibody response, and that's not going to be really great because you have no memory cells. But while that's going on, your body is making both T and B memory cells, so that the next time you're exposed to that same antigen, those memory cells are going to cause a more rapid antibody production response, which is also much, much higher than the primary response. So the secondary response is due to the fact that you have memory cells from the primary exposure that can then cause the production of antibodies to be much, much quicker the second, third, or fourth time you see that specific antigen. That brings us to the classifications of immunity. Immunity can be either naturally acquired or it can be artificially acquired. Naturally acquired immunity means that the immunity occurred without any endogenous substances being given. Naturally required active immunity is basically being exposed to some foreign material or pathogen and developing immune cells so that the next time you're exposed to it, you have a secondary response. For example, years ago, when chickenpox vaccine was not available, when someone got chickenpox, they would have the infection, it would cause the itchy rash and the fever, and over time your body would develop memory cells to that, and you would develop immunity to it so that the next time you were exposed, you would have a much more rapid response and you would not get that disease because you would be able to fight it off before it tended to grow. So an active immunity means that you developed the antibodies by exposure to a pathogen. Artificially acquired active immunity means that you are still going through that process, but instead of actively being exposed to a disease and developing antibodies to it, you're actually given a injection, usually, of a vaccine containing a weakened or dead or components of a foreign material, fooling your body's immune system into thinking you have that infection and therefore developing memory cells so that you don't get it. For example, nowadays we give a chickenpox vaccine to most children, which basically fools the children's immune system into thinking they've had exposure to chickenpox. So they go through all the steps of activating T cells, releasing cytokines, activating B cells, developing memory cells, so that the next time they're exposed to chickenpox, they will have that rapid secondary response and they will basically survive it. Artificially acquired passive immunity. Now, passive immunity is basically what happens when instead of going through the steps of identifying a foreign material, activating T cells, activating B cells, and producing antibodies. Passive immunity means having a jar of pre-made antibodies to a specific foreign substance, pulling it off the shelf, and giving it to somebody. Artificially acquired passive immunity is done when you don't have time to naturally acquire it. For example, if we go back to this response here, if someone is bitten by an eastern diamondback rattlesnake, that venom will cause serious problems in their body. They will go through the steps of developing an antibody response, but it'll take 3 weeks to get a response that is suboptimal because you're only making this many antibodies, and you'll be dead by then. So what do we do for that? When someone's bitten by an eastern diamondback rattlesnake, we pull off a bottle of antibodies that have been identified and put into a vial, and we inject the antibodies directly into it. So the antibodies directly attack the antigen, but you didn't make them actively. They're being given passive. Artificially acquired passive immunity means the giving of antibodies that you have in a jar that you've made because you don't have time to build up active immunity. What kind of things? Life-threatening conditions. rattlesnake bites, rabies, things that if you try and wait for your body's immune system to work, you'll be dead by that time. So it's artificial because you're using an artificial substance, but it's passive immunity because you are not actually going through the steps of producing active T cells, producing active B cells, and producing antibodies that way. You're just giving an injection of antibodies you got from somewhere else. And it's important for life-threatening conditions where you don't have time to build up immunity either naturally or artificially. There's also such a thing as naturally acquired passive immunity. What does that mean? Well, that means that you're getting antibodies naturally, but they're not your antibodies. When does that happen? Well, largely the only way that could happen is when a fetus receives antibodies through the placenta, and the mother has antibodies to some foreign material. So, for example, that's the reason why we almost never see a child under 1 year of age with strep throat. Almost never. Because of the fact that when a baby is born, some of mommy's antibodies to strep, and most adults become immune to strep, have passed through the placenta and are now circulating in the baby's blood. Now, they'll break down over time, but they'll be there for a while, providing a short-term immunity for newborns. So it's naturally acquired because it was acquired naturally from mommy to baby, but it's passive immunity because the baby didn't go through the process of building up the the activated T and B cells, the memory cells, and so forth. Allergic reactions. A couple of things about allergic reactions. They're divided up into 4 groups. The first group is probably type 1s are the most serious. These are what are called immediate reactions or acute anaphylactic reactions. These involve binding of antigens, specific antigens, to mast cells or basophils who have IgE antibodies on their surface. Remember, we said those are the antibodies on the surface of the mast cells. And when that happens, those mast cells then degranulate, releasing histamine and leukotrienes, as well as some prostaglandins that have various effects, depending on where they are and when it happens. This is an example showing how this works. Here is a mast cell. Wait a minute, no. Here is a B lymphocyte exposed to an antigen. It produces antibodies, and then those antibodies, some of them basically insert themselves as IgE antibodies on the surface of the mast cell. So now this mast cell sits there with these specific IgEs on their surface. For example, in my nasal passages, I have mast cells with IgE receptors to freshly cut grass antigen. And they're sitting in there, and because classes have been canceled for the rest of the semester, you will not get to experience this. But when the grass starts to grow in the spring and they start to cut the grass, what will happen is when I inhale the allergen, which in this case would be freshly cut grass antigen, will bind to those mast cells in my nasal passage, causing those mast cells to release histamine, causing those mast cells to cause vasodilation, increasing edema of the nasal passage, increased mucus production, and all the signs associated with seasonal allergies. What do you take for those? You take antihistamines to block the release of the histamine from the mast cells. So on a very non-life-threatening source, you can have these mast cells, like I said, sitting in your nasal passage waiting for a allergen to come by. But you can also have these on basophils floating through your bloodstream with specific IgE antibodies to, let's say, penicillin on them. And if you have that and a penicillin molecule binds to that, it's going to degranulate, releasing this into your bloodstream, which is going to cause all of the signs and symptoms of an acute allergic reaction. The low blood pressure, the closing up of the airways, all of the things associated with a life-threatening allergic reaction can be produced because of binding of a specific allergen to those IgE antibodies on the surface of those mast cells and basophils. Type 2 allergic reactions generally are transfusion reactions. This was, for example, if you give the wrong blood to somebody. If you give somebody type A blood that has anti-A antigens, obviously that can cause clumping of that blood because of those antigens, and could be a potentially life-threatening problem, but it does take a little longer than the type 1s. Type 3 allergic reactions are what are called immune complexes. What that means is that in many cases, antibodies and antigens connect together in the bloodstream, and then they float around, and they can cause reactions just from the presence of them being there. I've met many people who say, I don't ever get the flu shot because every time I get the flu shot, I get the flu. And I always tell them, no, you don't get the flu. What happens is when you get an injection of flu vaccine, you develop an immune complex reaction, which usually takes several hours to develop, and you will get achiness and sore throat and myalgias and things like that due to a type 3 reaction, but you're not getting the flu. And if you've ever had the flu, you would know that that's the case. And then the type 4 allergic reactions are what are called the delayed reactions. These are T-cell mediated reactions associated with the skin and associated to specific substances that get on the skin, such as poison ivy, which basically causes an activation of the T-cells and the abnormal skin changes associated with that. It's usually a very localized problem where the exposure occurred. It's not a systemic problem that can affect large parts of the body. A couple of words on transplantation and tissue rejection. This is a short list. There are many other tissues that have been successfully implanted since I made this list up. But suffice it to say, transplanting tissues or organs can be certainly life-threatening, life-saving for the individual with those problems. But to transplant tissues or organs, you have to be careful about the body's immune system, both of the recipient. You have to match the major histocompatibility antigens as close as you can to avoid an immune reaction. And if it isn't a perfect match like you would get from either the individual themselves or an identical twin, you'll have to be on immunosuppressive drugs for the rest of your life so that you don't mount an immune response to that transplanted organ or substance. Different types of transplants, what's called an isograft. An isograft is a graft from an identical twin. This doesn't happen very often, but if you do have an identical twin, your major histocompatibility markers are the same as your twins, so you can donate tissues directly to the twin and vice versa without any risk of upsetting the immune system. Autograft. Autograft is grafting to yourself or transplanting to yourself. When does that happen? That happens in burn victims. Burn victims will have large areas of skin that are burned, and what they have the ability to do is actually transplant skin from one part that's non-burned to the burned area, let them both regrow, and then transplant again as the skin that was replaced, that was removed, is replaced. So an autograft is a transplant from yourself to yourself, and it does not require any anti-rejection medications. An allograft is what you normally think of when you get a transplant. This is getting a liver or a kidney from somebody, or a heart transplant. And again, you have to have the closest HLA match you can, so that the recipient's immune system doesn't reject the organ. And even still, this individual is going to have to be on immunosuppressive medications for the rest of their life. And then xenografts are actually grafts from different species. A lot of people who have heart valve replacements, interestingly, we found that pig heart valves are very non-antigenic. Your immune system doesn't seem to mind them as much as other animals. So a lot of times when they do heart valve replacements, they will do valve replacements from pig valves, which seem to be fairly well tolerated. And lastly, autoimmunity. Autoimmunity I've hinted to several times so far. This is basically when your immune system goes wild and starts making antibodies and an immune reaction to your body's normal cells. There are many, many, many diseases that are autoimmune in nature, and the underlying cause of all of these are basically your body's immune system creating antibodies against your body's cells. Couple of examples we talked about in the endocrine system. We talked about Graves' disease, where antibodies that are made actually bind to the thyroid follicle cells and fool the thyroid follicle cells into thinking that they're being stimulated by thyroid stimulating hormone, and it causes overactivity of the thyroid gland. Type 1 diabetes mellitus is a disease in which you make antibodies to your pancreatic beta islet cells. Those are the cells that respond to high blood sugars by producing insulin. Once those cells are destroyed, they no longer make insulin, and causes the condition found in type 1 diabetes. Multiple sclerosis, which is a demyelinating disease of the brain. The myelin sheaths around those axons in the white matter of the brain are destroyed by antibodies. There's other factors in play there. We already talked about myasthenia gravis with autoantibodies against the receptors for the acetylcholine on the skeletal muscle fiber. Rheumatoid arthritis, very, very common debilitating condition where you make autoantibodies to your synovial cells of your synovial joint. Systemic lupus. Lupus actually is a disease in which you make antibodies to the nuclei of your connective tissue cells, causing the conditions associated with that. And then ulcerative colitis, which is an inflammatory autoimmune condition affecting antibodies affecting the cells of the colon. That concludes this part 2 of this lecture on the lymphatic system and immunity. Please let me know by email if you have any questions.